In the spring of 2003, the Sahara not as a barrier but as a corridor. Between roughly 14,500 and 5,000 years ago, the desert was something unrecognizable to anyone who has seen it today. Monsoon rains pushed north. Rivers ran across terrain that is now cracked and empty. Grasslands spread where nothing but sand moves now. Hippos wallowed in permanent lakes in regions where rain is now measured in millimeters per decade. Thousands of painted and engraved images across the Tadrart Caucasus show cattle herders, women grinding grain, children near water, animals that require green landscape to survive. The logical conclusion seemed obvious. A greened Sahara would have functioned as a bridge. Sub-Saharan populations moved north. Mediterranean populations moved south. Genes mixed. The green Sahara, in the dominant scientific framing, was a highway connecting two halves of the continent that had been cut off from each other. The evidence seemed solid. Pottery from Saharan sites matched styles found in the Nile Valley and in the Sahel, hundreds of kilometers in either direction. Stone tool traditions blended across the interior in ways consistent with people moving through. Cattle and goat bones appeared at sites where only wild game had been hunted before, which fitted neatly with the idea of herding communities pushing north from the Sahel as the rains arrived. Physical anthropologists studying skeletal remains noted features they associated with population mixing. The model was taught in universities, published in textbooks, and treated as the settled framework for how this region was populated. There was one thing nobody had done. Nobody had read the DNA. The gap had a logical explanation, a chain of assumptions, each one sensible on its own, that together pointed in the wrong direction. Ancient DNA work is a science of cold places. Permafrost in Siberia, limestone caves in northern Europe, high-altitude ice fields in the Andes. The Sahara offers the opposite of every one of those conditions. Daytime temperatures exceed 50 degrees Celsius. Humidity is negligible. Ultraviolet radiation destroys organic molecules relentlessly. DNA degrades in heat, and the Sahara is one of the hottest landscapes on Earth. Geneticists had ample reason to assume anything buried there would be too fragmented to read, and for decades, they largely did not try. The archaeological evidence added a second reason to stay confident without the genetics. When pottery from the Nile Valley appears in a Saharan campsite, the simplest interpretation is that someone carried it there. When cattle bones appear where only wild game was hunted before, the simplest explanation is that herders migrated through. Archaeology is extraordinarily good at tracking objects, but it cannot track bloodlines. For most of the 20th century, no methodology existed that could distinguish between a pot that traveled and a person who carried it. A third problem compounded both of those. North Africa remains one of the most poorly sampled regions on Earth for ancient genomics. By the early 2000s, Europe had hundreds of ancient genomes. The Near East had dozens. Between the Mediterranean coast and the Sahel, there was almost nothing. Without data, there is no pattern to question. Without a competing signal, there is no reason to doubt the existing model. Every assumption behind that model was defensible. Together, they produced a conclusion that the DNA would dismantle. The Takarkori rock shelter is the reason anyone one found out, and the reason is geological rather than intentional. The rock shelter is not exposed desert. It is a deep overhang carved into a mountain range, shielded from direct sun and insulated from extreme temperature swings. The women had been buried in dry sand that functioned by geological accident as a natural desiccation chamber, skin dehydrated before bacteria could consume it. Inside the densest tissues, fragments of 7,000-year-old genetic material had survived. Every previous attempt to extract ancient DNA from open Saharan burial contexts had returned nothing usable. The overhang had done what open desert could not, held the conditions stable long enough for something to survive. In 2019, researchers managed to extract mitochondrial DNA from both women. That particular molecule travels in